Today I'm honored to introduce CBD2, a functional biomarker database for colorectal cancer. The rapidly evolving landscape of biomarkers for colorectal cancer, CRC, necessitates an integrative, updated repository. In response, we constructed the Colorectal Cancer Biomarker Database, CBD, which collected and displayed the curated biomedicine information for 870 CRC biomarkers in previous study. Building on CBD, we have now developed CBD2, including a substantial number of newly discovered biomarkers, as well as non-biomarkers, and incorporates network visualization and topology analysis functions. Here are the highlights. 1. CBD2 integrates biomarkers derived from diverse biological sources and clinical applications, employing a standardized approach for annotation and classification. 2. CBD2 facilitates precision medicine in colorectal cancer by providing abundant data, including drug target information and visualized network analysis functions. 3. With its user-friendly interface, CBD2 aids in the identification and development of biomarkers for colorectal cancer. This is the pipeline of CBD2. We manually collected and classified validated biomarkers and non-biomarkers by manually reading the literature, and added innovative network construction and analysis functions. Compared with CBD, CBD2 updates 1569 biomarkers, bringing the total number of biomarkers to 2439. Simple guide on how to use the functions available in CBD2. Figure A shows 3 different ways offered by us to retrieve data: categorical search, keyword search, and advanced search. Figure B displays the steps and schematic diagram of the network analysis function module. To showcase the clinical relevance of the identified CRC biomarkers for diagnosis, treatment, and prognosis, we presented a case study that demonstrates the utility of CBD2 for analyzing biomarkers in a practical setting. We constructed the BBI networks for the existing diagnostic, therapeutic, and prognostic protein biomarkers in CBD2, supporting information, figure S1, and calculated network parameters in the Explore page, followed by gene ontology, GO. Functional enrichment analysis and KEGG pathway enrichment analysis, supporting information, figure S2. According to the box plot of topological parameters, we noticed that the closeness parameter had no obvious intergroup difference, while for betweenness and degree, the difference mainly appears in the prognosis group and the other 2 marker groups. Several biomarkers have undergone extensive investigation across multiple research endeavors. We searched CBD2 for collected p53 related information and found that almost all the research related to it focused on prognosis. Across these investigations, a consistent observation was evident: patients with p53 positive expression consistently faced a higher recurrence rate in comparison to those with p53 negative status. This trend invariably translated to a notably reduced survival rate for the latter group. 374 ncRNA biomarkers have been collected in CBD2, of which 210 belong to mRNA. We construct a network elucidating the interactions between ncRNA and genes. In the ncRNA gene interaction network, we found 7 of the hubs are included in CBD2. We additionally conducted enrichment analysis utilizing both KEGG and DisGENET on the delineated network, the results of which exhibited a pronounced correlation with CRC, underscoring the potential significance of the identified ncRNA gene interactions in the context of this malignancy. Summary. In this study, we have updated the CRC biomarker database, CBD2, which have included 2439 reported biomarkers for CRC. We have added drug target information and biomarker-biomarker interaction, BBI, network analysis function in this update. We have provided a user-friendly interface, and we anticipate that the updated version of CBD2 will serve as a flexible and valuable tool for advancing CRC biomarker research. Thank you for listening.